I'm excited to introduce you today to Miromatrix, which is kind of exciting because we're going to talk a little bit more about tissue engineering as well and what we're able to do on that. At a high level, our goal at Miromatrix is to eliminate the organ transplant waiting list by bioengineering transplantable organs. And the way that we do that is our technology is perfusion decellularization. That's our ability to actually take a whole organ, perfuse a mild detergent through it, and remove all the cellular material. What that allows us to do is really take what nature already created and have that perfect scaffold to then move into our patented recellularization, where now we're able to place that in a bioreactor, perfuse cells back into it, now to be able to reanimate and create transplantable organs. At a high level, just some quick history. We were founded in 2009. Our IP is issued worldwide. Internally, and what I'll talk a little bit about today is mostly on our liver side. We have two main whole organ programs, led by the bioengineering of our liver program, large market opportunity, but there's really multiple reasons why liver is our lead organ. One, and most importantly, is from a patient standpoint, and that is there's no alternative therapies for end-stage liver failure patients, right? There's no drugs, there's no devices, there's no dialysis. So we're really looking at a patient population that needs solutions. We see this as a rapid pathway to market because of that as well, with no alternative therapies. Our second overall program is a transplantable kidney, with over 100,000 patients today waiting for a transplantable kidney. There's no lack of need in that area as well in terms of something for therapy, and it's a very large market opportunity. And then we are also working on the bioengineered whole heart with the Texas Heart. But just as important as our whole organ, and I won't get into this today because of time, it was very important as an early stage biotech company to demonstrate that we could also manufacture stuff and that we could control our manufacturing process. So we've actually commercialized and launched Miromesh, which is for hernia repair, and Miroderm, which is a wound care product. This is a porcine liver-derived product where we decellularize the whole liver, and now we're able to create that new surgical product. And from that standpoint as well, a wound care product. One of the key caveats of this, and I'll show you this in the next slide real quick, is just the overall notion from that was a regulatory standpoint, because the open question still is, as you take the whole organ and decellularize it, you could start with a human organ or you could start with a pig organ. If you start with a pig organ, is there going to be an immune response? How does the FDA and others feel about that? By bringing these through the 510(k) process and getting them on the market, we've essentially de-risked that. We've been able to have that now implanted in thousands of patients with great clinical data associated with it. And I won't get into that. We do have 2 active clinical studies to drive adoption and revenue associated with that. 250 patient multi-perspective studies that'll be concluding this year, and data will be coming out later on this year on that as well. But just as real quickly, as we think about that market strategy and go to market, how do we see ourselves bringing whole organs all the way to the market? It really starts with first demonstrating that perfusion decellularization is effective. I just described our commercial products. We commercialize that ourselves. We've been audited, the facility's been audited by the FDA. Those products are available today. So we've achieved that. Recellularization, that's the ability now to put cells back into the matrix and demonstrate that we can reanimate it. And really which we've got great data on that as well. I'll show you a little bit just briefly in a second here. But the event that we're gearing up for this year is more of the pivotal preclinical study, and that's to bioengineer a liver inside of our lab, place that back inside of a large animal, inside of a pig, take out the native liver, and demonstrate that pig can survive. That's targeted for this year, and that data to be released later on this year. The studies are going really well on that front. Once we're able to achieve that, that moves us into the IND, which I'll describe in a second. But if we go ahead and play this video, this is actually a re-endothelialized liver that's being transplanted back inside of a pig. You can see we took off that bottom clamp. So this is now being perfused with the host blood at this point. You can see the liver priming very well. You're going to see a white strip on the top up there, and that's actually where we don't have the circuit completed. Now when we take that off the hepatic vein there, at this point now we have native blood perfusing through our graft. So this is a graft that's been re-endothelialized and put back in. If you look at that, it looks just like a liver, right? So that's the advantage of starting with that scaffold and being able to take that ability of starting with what nature already created. If we put this together, so what does this look like in timing? As I mentioned, we're targeting that first pig rescue by the middle of this year. Once we're able to achieve that and publish on that, that then leads us into repeating other studies to then support our IND submission, targeting an IND submission. And again, what we're targeting for this, as we see this moving rapidly, is as early as 2020 to be first in humans. And then because of the nature of the product, we'd be targeting an orphan status associated with that to really fast track that as well, with the potential to even be a product by the end of 2022. So that's a quick introduction to Miromatrix, kind of high level, our technology, what we're doing, and our path forward.
So you got some great stuff you're working on here, Jeff. Let's start with the biology. And obviously, it's a pretty complex problem that you're trying to solve. So what would you say are some of the key biological processes breakthroughs that you guys have been able to achieve?
I think one of the key ones is that vasculature. So when I showed that video, and I actually showed it being anastomosed back inside of the animal, and then being able to perfuse with the pig's blood back through it without thrombosis, that was one of the key breakthroughs. We spent about 3 years optimizing that process and demonstrating that we can get functional vasculature to the point that we can now transplant these back into an animal model without heparin or any antiplatelet therapies and have that long-term sustained perfusion of the graft. We're actually getting ready to publish that, and that should come out in about 3 or 4 months.
Are you having to do anything special to manage any stenosis related to the anastomosis?
No. I mean, what we've seen is really targeting to demonstrate that you have great endothelial coverage. And we put a lot into that. And I think that's the value of commercializing our early products as well, is understanding the quality system, understanding release criteria. So what we've looked at is non-destructive markers to say, when is that graft ready? When can it be predictive that you're going to have in vivo success?
And beyond, just the vascular part of it, what else are you doing that, kind of enhances the probability that the tissue survives the engraftment?
Yeah. I mean, the vasculature was the biggest one in terms of if you don't have sustained vasculature and you're not able to support the cells that you put back into that, there's really no way to sustain that organ long term. So that was key. And the second one that we look at now is as we put in the hepatocytes and the cholangiocytes in the liver, or we're recapitulating the nephron inside the kidney, is just what does that growth profile look like? So we do spend a lot of time in the manufacturing standpoint of looking at when is that graft ready to make sure your cells don't overgrow or undergrow.
So as you move forward with your next pig study, talk about the design of that.
Yep. So for the pig study, it's very straightforward, like our end clinical studies will be for this as well once we get through the FDA. And that's the notion of actually taking that liver out of the animal. And at that point, an animal can only survive about 24 hours without its native liver. So it's a very defined study. We're actually going to take that liver, explant the liver out, put ours in, and then study how long that animal can now survive with our bioengineered liver. And it's slightly more complex in the sense that we're not creating a liver back for a pig. We're actually re-endothelializing or putting vascular cells in there human. So we're actually putting human cells back in and then transplanting that back into the pig. So some of the challenges that we have there is getting the immunosuppression right to make sure that we can overcome that cross-species barrier. But we've done that on the vascular side, and we're confident we can do that with the fully recellularized liver too.
So what type of results would you judge as a success such that you would move forward in humans?
So early on, a success is as little as a couple of days or 7 days survival rate inside the pig we see as a great success. Again, because we have human cells going back in, that sets up the stage from a market endpoint of even a bridge to transplant. But if you look at it from longer term, 7 days is that great foothold. After that, we'd be repeating that and wanting to go out 30 days. We feel once we're to 30 days and have continuous perfusion and functionality, that should be predictive of long-term success.
Okay, so this is a study that, relatively speaking, you'll have results in a reasonably short amount of time.
Absolutely. We're actually growing the grafts at Miromatrix today, and they'll be transplanted back in with the Mayo Clinic over the next 2 to 3 months. So the transplant model's been worked out. We have all of our controls done, and now we'll be starting to put in our active grafts.
Okay, so assuming this is successful, what else do you need to do on the manufacturing side to be able to go into humans?
Yeah. So after this, I mean, this is a great proof of principle. After this, then we start gearing up on the manufacturing side in terms of cell banking, the endothelial cells, and getting those ready, and then the hepatocyte isolation to be able to support and recellularize those. I mean, the great thing about the liver as an initial target as well is end-stage liver failure patients, you only need about 20% overall liver function for the patient to be viable. A failing liver might be at 10, 12%. So you're not looking at a huge boost that you need. So what we're actually targeting is about a 20% overall functionality that we'd be giving back to the patient. Because the liver is the one organ that can regenerate itself as well, we're also targeting and looking at what that regrowth looks like inside of the patient once these are put back into them.
So, as you think about going into a human study, what would be your, not that these patients aren't in desperate need of a new liver, but what would be your ideal candidate in terms of the disease that had led to their liver failure?
Right.
Who do you want to avoid, and who would be ideal?
Yeah, I mean, unfortunately in this area, right, we have 40,000 patients in the U.S. a year that are dying of end-stage liver failure because they can't be one of the 7,000 that receive a transplantable organ. So the patient population's pretty well available in what we're looking at. What we're targeting for our first studies is end-stage liver failure, patients with about 6 months of life left, to be able to put that in, because we're going to be targeting 6 and 12-month endpoints inside of our clinical study. But as we design this and continue to look at it, I guess the fortunate but unfortunate part of it too is many of these patients, their mortality rate's very high within 6 months. So the clinical study also can have a very solid endpoint in terms of total mortality at a relatively early time point.
So, commercially, what do you think would be a sufficient amount of life for this type of graft that somebody would want to go through what's going to be a fairly complicated surgery to get that extra X number of weeks, months, years of liver functionality?
I mean, I think it's best to talk to the patients and get their perspective on that at some point. But some of the limited patients we talk to, they'd be happy with months compared to where they are as they're facing end-of-life decision. But our goal is really to have that long-term solution. The data that I've seen so far and the data that we continue to generate, and if we look at whole organ kind of things, once you get past that 6-month hurdle or so, we don't see a lot of reasons why the graft should fail longer term besides graft failure from an immune response standpoint. But from the design of it and the tissue engineering and the functionality of it, we're really targeting a long-term solution. So our end goal is the patients get a liver, and once you get over that initial barrier that you would do with any type of transplant, that should be your long-term solution at that point.
Okay, and just to touch on your wound care product, talk about that a little bit. I know it's not the primary focus of the company, but it can generate some revenue. What's the commercial plan for that?
Yeah, so we're really excited about that. As I mentioned, that's a 510K. We've developed that. The blessing and the curse with the 510K is you can get it through the FDA, but you don't require clinical data. To really get the uptake of that product and sales, we invested in a clinical study. So we're wrapping up right now a 50-patient multi-center clinical study where we're actually targeting in that diabetic foot ulcers. And there's many advanced biologic products that target that diabetic foot ulcer, right? Because if that patient comes in, they get standard of care for 30 days. If it doesn't close, then they get these advanced biologic products. And the challenge, you're then in a race, because if you can't get that DFU closed, that could lead to amputation, and that's a higher mortality rate associated with it. So what we've really targeted is, inside of our clinical study, is not taking those DFUs when they first become chronic. We decided to take the DFUs that all the other products couldn't close. So let's go after the hardest to close wounds to try to show that liver-derived tissue is different in terms of what it can do to the wound care response and what it can do to those wounds. So that's the design of our clinical study, where we actually enroll patients who have failed other advanced biologics. We just published on this a case series in wounds in October where we demonstrated that we could close over 50% of those patients. We just are at 50 patients in our clinical study as of yesterday, so we hit that milestone, and we're looking to have that data out by the end of this year as well. And I can tell you that it's trending very similar to what we've seen in our published case series. So we're excited about it. So the product's for sale today. We're selling it through independent distribution. We brought on our first direct rep a year ago who has put up tremendous numbers in the first year, just demonstrating the value of the product and the ability of it. And what we're looking to do short term is continue to develop that sales force and grow that product line in conjunction to what we're doing on the whole organ.
All right, well, great.
Thank you. Thank you for the question.